Hey there everyone. This video is going to take you through the remainder of the notes that you need for this particular part of Module 2, Exploring and Analyzing Biodiversity. We already took a lot of these notes. I'm going to kind of loop you into where you should have ended up. So hopefully this stuff looks familiar to you. These PowerPoints are also on our Canvas site, so you have access to those. So let me go ahead and get started with this. So you should already have a section in here where we're talking about quantifying biodiversity. We talked about the difference between species richness and species evenness. Species richness being how many different types of species are present. So again, if you're on the savannah, lions and zebras and hyenas and elephants and rhinoceros and hyena, those are all the animals, and then the different types of grasses there are there. And then species evenness, which is the number of individuals in each species. And remember, we did this comparison, which showed us that just because you have the same species richness doesn't tell you the full picture. You need to look at the evenness as well. Now, usually when you have a more even ecosystem, it's going to be more stable. But there are times when this isn't natural. So again, when we find out data, it doesn't necessarily tell us just one thing. It's just a way for us to evaluate what we're looking at. So this is where some of you may have stopped. How precisely do we know how many organisms there are in a certain area? Nobody's sitting there and counting every single blade of grass or every single zebra. One method is called quadrat counts. Quad means 4 sides, so a quadrat is a way to measure biodiversity in terms of richness and evenness. The procedure: mark out the overall area to be counted, select quadrats in that area. A quadrat is literally a square, and then you perform the count and then use math to estimate the total biodiversity of the area. The quadrats can be of all sorts of different sizes. They can be small, they can be large, it just depends. So how precisely does this work? If you look over here, you notice you map out your entire area, and then you select sample quadrats. And you can see that there are different ways of doing this. We are going to be doing the random one. When you do this activity in class, you are going to have a random selection of quadrats that you're going to have to count and then process, and your quadrats are going to be different than anybody else's in the class. One of the things to consider when you're doing this is, okay, when you literally make this out of pipe or wood and you throw it down on the ground. Once you map out where you're going to be doing your counts, you need to figure out exactly how you're going to count things, because not everything is going to be in the lines for you. This is pretty much what we're going to follow, which is if half or more of it is in the square, you're going to count it. But if less than half is in, if more than half is out, then you don't count it. So keep this in mind for when we do the quadrat count in class. So you need to make note in your notebook of what kind of organisms quadrats are good for, and then what kind of organisms probably won't do well with a quadrat count. Like, let's look back at this. Think about crabs. Think about what they do, especially when humans come near. You know how they scatter? Probably quadrats are not going to be the best way to count these guys. Quadrats usually work best for things that stay put. So think about grasses and flowers and trees, and if you're out in the ocean, anything that kind of stays on the ocean floor. So organisms associated with a coral reef, not the fish, but clams and sea urchins and sand dollars. Those things move, but very slowly. Now, if you do have things that move a little bit more quickly, like let's say bugs, what a lot of people will do is they'll put a quadrat down and then they'll quickly take a picture, and then they'll do their count from the picture. And another nice part about that is that you aren't going to harm the organisms, because you're kind of in their territory for a very short amount of time, in their space for a very short amount of time. So quadrats are usually good for things that move slowly or not at all. This is not going to do well with things that move quick or things that are really big. Like you're not going to be able to throw a quadrat over a herd of elephants or whatever. So there are other methods of counting them that we're not going to get into. We're just going to go ahead and stick with this one. Okay, so here you can see a quadrat count that's being done. I will, during the activity, actually explain the math associated with this, but basically what you do is you count what's in these random samples. Each one of those is a quadrat in this larger area, and then you divide it by the area that's in there, and then you kind of use that number to estimate the total count, and you're going to see you can actually get pretty close with that estimation. So that is the end of this particular series of notes. So go ahead and look for that second video on, basically it's on evolution. So thanks for listening.